Alright guys, today we are going to talk about Lac Operon. Now, we have discussed this thing before as well, that when a sperm joins with the ovum, the new cell formed is called what? Zygote. Now, we have also learned that this zygote is actually a totipotent cell. Why do we call it as totipotent? Because all the genes are on and active. All the genes, they can be transcribed, and all the genes, they can produce the proteins which are required for a cell. But once this zygote, it reached to 16 cell stage embryo. Now what does this mean? Please pay attention. Let's suppose that this is zygote. First division. From one cell, two cells are made. This division is called cleavage. Second division. 1, 2, 3, 4 cells are made. Third division. 8 cells are made. Right? And when there will be fourth cell division, and which cell division I am talking about? Mitosis. When fourth time in the embryo mitosis will take place, from one cell, 16 cells will be formed. At this cell stage, the cells, they do not remain totipotent anymore. They lose their totipotency. They become pluripotent now. Because now the cells, they start becoming specialized. Now all the genes are not active, only certain genes, they will remain active according to the cells which they are going to be specialized and which tissue they are going to make. Now just you see, not related to this topic, but you guys know the identical twins, right? Now before 16 cell or at the 16 cell stage, if a cell separates from it, from this embryo, that will change into a complete new embryo, and that will be identical. After 16 cell stage, if a cell separates, it cannot develop into a complete embryo because cells are not totipotent anymore. Understand? So up till 16 cell stage, if a cell separates, and no one knows why do these cells separate. Some say by accident, by falling, mother falls down or some jerk happens, right? Because of that, these cells separate, and what happens? An identical twin is formed, right? Because till 16 cell stage, the cells are totipotent. When they lose their totipotency, at the fifth cell division, there is no chance of becoming an identical twin. Now, question is, why the cells lose their totipotency? Why not all the genes remain active? If all the genes will remain active, it means every cell will be able to produce the proteins which are not even required in that cell. So that will be the wastage of resources and wastage of energy as well, right? So these cells, they become turned off, which are not required in a cell. And the cells which remain turned on, because they are required in that specialized cells, and even the cells even the genes that remain active, they only produce the product when it is required. When it's not required, they do not work. To understand this turning on and turning off a gene, we are going to take the example of Lac Operon. Now, this over here, we have a bacterium. We know that outermost covering of the bacterium is what? Cell wall. Inner to cell, we have cell membrane. Then we have plasmid over here, and we have linear DNA. We also call it as loop of DNA, or we also call it as chromosomal DNA, right? One very important thing which we have learned before, that the bacterial DNA does not have introns. Now question is, what are introns? Now introns, these are actually what? Non-coding part of DNA. Now I have taught you last year the structure of chromosomes. Do you remember this or not? Now there are nucleosomes, right? Now nucleosomes are histone proteins on which what is done? Wrapping of DNA is happened, right? Do you remember this or not? And in between two nucleosomes, the DNA is called what? Who remembers? Let's suppose that this is one histone protein, right? And look here, I have told that there are 2 times wrapping of DNA. Now this whole DNA and histone proteins collectively called nucleosome. This is one nucleosome. This is second nucleosome. Now this part of the DNA from here till here, which is not wrapped around the histone proteins, what is this called? Intron. This is intron, but it has a name. This is called linker DNA. Remember I told you this. What is this called? Linker DNA. And this part of the DNA is non-coding part. This part of the DNA is what? Non-coding part. Now remember that bacterial DNA do not have introns. Their DNA is all coding. Their DNA is all coding. Next thing was that this bacterium, the diagram that I drew, this experiment and the study about Lac Operon, it was done on, you have learned before, called E. coli. The full name of this bacterium is Escherichia coli. Now some people, they pronounce it as Escherichia coli. Wrong pronunciation, right? Correct pronunciation is Escherichia coli, right? Now, we can feed this bacterium different kind of nutrients, right? Now, we can take a petri dish. Now, in the petri dish, over here we can have nutrient medium. This nutrient medium can have glucose in it, right? Let's suppose that we take another petri dish. In this petri dish, the nutrient medium it contains lactose. We have 2 petri dishes. In one petri dish, the nutrient medium is what? Glucose. The other petri dish, the nutrient medium is lactose. Now, when the glucose is there, should lactase be produced over here? No. If the genes present inside this Escherichia coli, they are, you see, producing lactase when the nutrient medium is glucose, what it will be? Wastage of energy, wastage of what? Resources. So the lactase enzyme should only be produced where? In this nutrient medium. In this nutrient medium, that gene will remain turned off. Right? Now, how this gene turn on and how it turns off, that is what we are going to learn. For that purpose, what we are going to study, first of all, the genes and their products that are responsible for making the products that will help in digestion of the lactose. Now, first of all, I am going to draw a complete this operon. first of all, I'm labeling this one as P1. This P1, it is representing promoter 1. This is a sequence of genes I'm going to represent, right? Now this is the promoter. You guys have learned that every gene has a promoter region. Last year, when we learned about the transcription, at that time I taught you. And we learned that there are 2 types of promoter regions over there. Who remembers? Just raise your hands. The boys who were absent yesterday, they try to answer. Sahib Zala, do you remember? Hamza. There were 2 types of promoter regions which we discussed last year. Operator is something else. Promoter is something else. Promoter is the place where RNA comes and binds. What is this? Tata. Uncle ko Tata bolo when he's going. when uncle comes or aunt comes, right? That say Tata to uncle when he's leaving outside. Tata box. Remember? Now, this is now Tata box. Let me remind you guys. What is this? This is a sequence of nucleotides that has TA, 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 TA. Repeat of TA, 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 TA. Thymine, adenine, thymine, adenine in the DNA I'm talking about. In the DNA I'm talking about. Second, this is called Tata box. That is the repeat of TA, TA, TA. Right? It can be different number over here. Second is called Pribnow box. The Pribnow box is repeat of adenine, thymine. Atta box. Not Tata. This is called, this is Tata box, right? And this is Pribnow box. So, this is the promoter region of the gene. Now, which gene? Gene I. Now, what is this gene? Let me tell you. This gene is called as inducer gene. What is this called gene? Inducer gene. Then, after gene 1, we have P2. Now, this P2, it represents promoter 2. Now, this P1 was the promoter for gene I. Right? Now, this P2 is the promoter of other genes which are in a sequence. And they are responsible for digestion of lactose. Now, after P2, there is O region. O refers to operator region of gene. Understand? After that, there are 3 genes. This is called Lac Z, then Lac Y, and then Lac A. Why Lac? From lactose. The Lac word is representing lactose. Now please pay attention. This Lac Z enzyme, it produces, the Lac Z gene, it produces an enzyme beta galactosidase enzyme, right? This Lac Y gene, it produces an enzyme called permease, and Lac A gene, it produces an enzyme called as transacetylase, right? Now, why this is called beta galactosidase? Because of the presence of glycosidic bond in between the lactose molecule. There is glucose and galactose present, right? So that is that present over there. Now, obviously, they will not directly produce these enzymes. Obviously, transcription will take place. Messenger RNA will be formed, which will be translated into these enzymes. Now, there can be two situations. Situation number one over here. When there is glucose in the nutrient medium, when there is no lactose, right? And the situation number two, when there is lactose present in the nutrient medium. So first we are going to discuss if the lactose is not present in the nutrient medium, how the gene, these three genes, they will remain turned off. Understand? Because at that time, these three genes, they should not produce any messenger RNA, any enzyme should not be produced for digestion of lactose. Now, before we move forward, let me tell you that this enzyme, it helps in what? Breakdown of lactose. This permease enzyme, its job is to increase the permeability for the lactose. In this situation number two, if the lactose is present in the surrounding of the bacterium, more of it should be taken in, right? So the permease enzyme, what will it do? It increases permeability for lactose, right? This transacetylase. Now, the word trans is coming from transition, shifting, right? So what does this enzyme do? It shifts the acetyl group from one molecule to another molecule. We are not going into the chemical detail. We are not going into the chemical detail right now. Now, again, first we are going to discuss when lactose is absent. The first scenario, right? Or the first situation. Again, let me draw this hole. Lac Operon. Again, what do we have over here? P1. This over here is gene I. Then what do we have? P2. Then we have operator region. Then we have Lac Z. Then we have Lac Y. And then we have Lac A. Understood, everyone? Now what will happen? Remember, lactose is absent. Lactose is absent. What will happen? That the RNA polymerase will come, will bind to the promoter region 1 of what? Of inducer gene. It will start the transcription and it will make what? Messenger RNA. So this is what? This is the messenger RNA of gene I. Understood? Next step, what will happen? With this messenger RNA, the ribosome, it will bind. So this is the ribosome. And the process of what will happen? Translation will take place. When the translation will take place, a protein will be formed. Now, please pay attention. The protein formed from the inducer gene, this protein is called as repressor protein. What is this protein called? Repressor protein. What happens? That this repressor protein, it goes and it binds to the operator region. This repressor protein, it goes and binds to the operator region like this. Now, this RNA polymerase, it will move forward and it will reach over here onto the P2 region, promoter region. But it can't move forward. Why? Because on the operator region, what is attached? Repressor protein. The word repressor express itself, what means? Suppressing, stopping, right? Speed breaker is there. A wall is there, right? So what will happen? When this repressor protein is bind to the operator region, RNA polymerase cannot move forward. No transcription will take place of Lac Z, Y and A. Gene is turned off. Gene is turned off. Understood everyone till here? Now, we are going to discuss what? Situation 2. When the lactose is present in the nutrient medium. Now, let's suppose that over here again, this is the second, that lactose is present. Again, what I'm going to do, whole Lac Operon I'm going to draw. Again, what is this? P1. What is this? Gene I. Then what do we have? P2. Promoter region 2. Then what do we have? Operator. Then what do we have? Lac Z. Then Lac Y. And then Lac A. Now remember that what has happened? With the operator region, this is the repressor protein which is already bind to it. The repressor protein is bind to DNA. The repressor protein is bind to DNA, blocking the passageway of what? RNA polymerase, right? Now, remember, lactose is present. Now, when the lactose is present, the lactose will enter into the bacterium. Let's suppose these are what? These are lactose molecules. Now, these lactose molecules, they will enter into the bacterium. Now, when these lactose molecules, they will enter into the bacterium, these lactose molecules, they will go and they bind to the repressor protein. Understand? So what happens that, let me mention over here, that lactose molecules enter the cell and bind to repressor protein. Now, when they will bind to the repressor protein, what will happen? The conformation, the shape of the repressor protein will change. The conformation or shape of the repressor protein will change. As the shape of the repressor protein will change, it cannot remain bind to the DNA molecule anymore. Why? Because before it was complementary. The shape of the protein was complementary to the DNA molecule. Now it's not complementary anymore. So what do we say? That what happens? That as the lactose binds to repressor protein, its conformation change and it's released from DNA. It becomes released from the DNA. Now, when it will become released from the DNA, let me quickly draw this again. This is P1, gene I, P2, O, Lac Z, Lac Y, and Lac A. Now remember that over here, RNA polymerase was already present there, but it could not move forward. But now what has happened, that this repressor protein is now released. The repressor protein is now what? Released. Let me show. And remember that with the repressor protein, lactose molecules are bind. Now there is no blockage in the front. There is no speed breaker in the front. There is no wall in the front, right? So this RNA polymerase will move forward. And with transcription, what will happen? Messenger RNA will be formed. And transcription will happen, and then translation will take place, and the proteins, these enzymes will be formed. Now what will happen? Just a minute. Now what will happen? That as the permease will be formed, will allow more and more entry of lactose, right? And these two enzymes, they will help in breakdown of the lactose. These two enzymes, their job is helping in breakdown of lactose, but this enzyme is only helping in intake. Got it? Now eventually what will happen? All the lactose present in the surroundings will come inside and will be used up. Now, when all the lactose is used up, there is no more lactose in the nutrient medium. Should this gene keep on transcribing? It should stop transcribing at that time. So at that time, what will happen? What I was talking about, that once this whole lactose is totally finished, right? Now what will happen? That the lactose which was attached to the repressor protein will also be released, so that it can be used up, right, in the system. So when it will be released, now repressor protein is what? Free, right? Now this repressor protein will again come what? And bind to the operator region. And another thing can also happen, what? That this P1 again can be transcribed, right? This gene I, which is the inducer gene, it will again be transcribed, and again it will go through this process of translation, right? And what will happen? Repressor protein will be formed, and this repressor protein will go and bind to the operator region. Now remember, repressor protein cannot bind to the operator region until or unless there is lactose present. When the lactose is absent, so the repressor protein is free to do whatever it wants to do, right? So at that time, repressor protein will go and bind to the operator region, and the genes, these genes, they will stop transcribing, and the genes, they will become turned off. Understood, everyone? So this is how the Lac Operon works. Someone had a question. Yes, Amir.
So when the operator region is blocked, so what happens to the mRNA? Does it just go back or?
Now, listen, remember, the messenger RNA which are already present in the cytoplasm, we have learned that they are eventually broken down back into nucleotides, right? And those nucleotides are reused for making this particular messenger RNA or some other messenger RNA. Understood? So there are nucleases There are nucleases that do break down, right? These you see these messenger RNAs. Understood?